Okay, well, good afternoon, everyone. We are going to cover our last little presentation regarding our exam. The exam on Thursday was going to cover bones, joints, and muscles. We've covered the chapter on bones. That was chapter 6. That also leaked a little bit of information from chapter 7, which in your Martini book is basically an atlas, name that bone. Then we covered chapter 8 last time, which is over joints and joint spaces. And then today we're covering 9 and 10, which are dealing with muscle. And there's just 3 presentations involved with today's, or on Thursday's, lecture exam. That will be a full-length exam. It'll be largely multiple choice. You will need your Scantron form. There will be a few fill-in-the-blank things. Certainly your vocabulary is fill-in-the-blank. There will be a case study. Likely someone's going to fall and break a bone. That shouldn't be hard to imagine. And there will be your connection questions. How is the skeletal system connected to other systems? And tell me specifically an example of how that connection is made, as well as 2 other systems for the muscular system, right? What are 2 other ways, what other systems are connected to the muscular system? There'll be a few PowerPoint images, nothing you haven't already seen. And vocabulary will go through 52. And I know there's a disconnect between my slides and your slides that I'm using right now, and I'll figure out what the problem is, but you're going to go through lunar, right? Lunar is the last term you have on your slide 52. So picking up with slide 50, let me go through the rest of the vocabulary. Then I'll entertain any questions or concerns you have related to the first couple of lectures, and then we'll finish up with muscle today. So lemma, we'll see this term today. It means a rind or a peel, an outer layer. Right, an outer layer, like the watermelon rind. And we'll see the sarcolemma today. There's also a neurolemma. Lepsy, seizure. Epi, we know, means on or upon. So epilepsy literally means seizure upon seizure upon seizure. Let, small. Platelets are small little fragments of cells that will be involved with blood clotting. We'll see that for exam 5. Leuco means white. Your white blood cells are also your leukocytes. And I think I've got this term twice in the mix, so it must be an important one. I think you'll find leuco labeled in here twice. Lig. You know that a ligament is something that binds a bone to another bone, right? Ligament. So what kind of tissue makes up ligaments? What kind of connective tissue forms ligaments? And tendons. Dense regular, right? Dense regular connective tissue. Lip, like lipids, fat. Logy or ology, the study of something. Here we are in a biology class learning about life. And lysis is to break up or dissolve. You've heard of lysosomes, those vesicles that dissolve things. Hemolysis, hemolysis would be the breaking down of blood cells. You've seen lateral, to the side. Leuco, there it is again by mistake. Lith, stone. If you have a kidney stone, they might put you in a lithotripsor. It's basically a sonicator. It's going to try to shake that stone down. So lith means stone. And lucid, we saw the stratum lucidum in the skin. It was that layer of clear, that clear layer that was found only in thick skin. And then, just to keep up with what you've got, lunar, right? Moon-shaped, or in the moon, referring to the moon. So that finishes up everything through 52 on your slides, correct? Did I miss anything along the way? Very good. So vocab went really well for many people. There were a few students I remember saying it did not go well for. I hope that it goes well for you this time. Any thoughts, concerns from the last 2 presentations over bones or joints? Yes, ma'am. You just copy and paste the link, and it should bring you right to YouTube, and it should just pop up. Show me afterwards, and we'll try it again. Okay. Has anybody else had trouble with the YouTube links? Well, you can look my name up on YouTube as well, right? You can do that, but also if you go on Blackboard, I've got this little site next to mastering. There's also another link that says YouTube video links. Click on that. I think I've got 2 folders, summer and this current fall. And then each lecture I add to that list through the fall. And I think they are loading from top to bottom. So at the very bottom, you'll see lecture 9. I think it was 9 was last time. And if you just cut and paste that link up into your browser and push go. It should bring you right to YouTube and pop that video up. Make sure you're not missing a letter or something. So we'll try it again. Okay. Some people find that very helpful, okay, to listen to the lectures again. It's another way. If you're looking for one more thing to do in your preparation, maybe if you've already started a study group or you're already doing all your mastering assignments and you're looking for one more thing you could do, one thing might be kind of straightforward, is just to re-listen to the lectures. And by doing so, have your notes out in front of you and just go through that process again. It's amazing how many more connections you will make the second time you hear it. So that would be a strong suggestion. Some of this you've seen. Again, we've already had muscle in lab. So some of this is going to look familiar to you. And you know that muscles are named based upon their Latin names, and that those names oftentimes have meaning if we understand the prefixes that are involved. So, for example, the rectus abdominis, right? Rectus means straight. So it's a straight muscle within the abdominal region. And don't forget that rectus abdominis ends in IS, right? We get a lot of US's there on the lab practical. So you want to be carefully looking at those letters, especially at the end of the muscle names. Other muscles like the biceps brachii, well, it's in the brachium, and there are two heads to it. There are actually two tendons that come up into the shoulder, one that goes up to the coracoid process, and one that goes up along the head of the humerus and the glenoid cavity. The triceps brachii has three such tendons, three heads to the muscle in the brachium. You're not responsible for knowing the different heads of the muscle. If you look in Amerman or in your atlas, you'll see it labeling, the lateral and the medial head of the biceps brachii. You don't need to worry about that. Just know the triceps or the biceps brachii. The tibialis anterior, for example, it has to be the muscle in front of the tibia, right? The name tells you that. Also, muscles come in four basic patterns. And the word here is fascicles. I'll describe that word to you in a few minutes, this word fascicle. But for now, just think, even from across the room, I can tell that muscles have four basic patterns of arrangement. Number one, some muscles are circular. You learned about two of them already, the orbicularis oculi, the orbicularis oris. Another word for circular muscles are sphincters. You have sphincters that are throughout your body, especially in your digestive system. And these circular muscle groups can close and open, thereby allowing certain molecules to pass through. Convergent, like the pectoralis major. If you picture that pectoralis major muscle, it's triangular in shape. And you've got fibers attached to the sternum, and then you've got all those fibers going to an attachment point in the armpit. It has a triangular shape to it. Those are convergent. Then there are parallel, where the muscle fibers and these fascicle arrangements are running parallel to each other, like the biceps brachii. And then there's pennate. And pennate refers to a feather-shaped muscle. Even from across the room, if you look at the rectus femoris, for example, you'll see that it has a feather-like arrangement. The tendon and the muscle fibers attach at an angle. Now, again, we're stuck with the Latin names. We've been using these now for over 120 years, pretty much, and there are a few variations on the theme, but for the most part, we're stuck with the Latin names. We did not have a levator or a profundus in our muscle list this semester, but we did have a quadriceps, right? We don't have the muscle, right? But we have the group, the quadriceps femoris. So you know that it's four muscles along the femur, right? The quadriceps femoris group. What are those four muscles? Name them off for me. The four muscles of the quadriceps femoris group are rectus femoris and the three vastuses, right? The vastus lateralis, intermedius, and medialis, right? Whichever one I didn't say, right? So medialis, lateralis, intermedius, and the rectus femoris. Make sure you know the four members of that group. So again, in this picture, you can see the arrangements. I'll start with the same order that I went through a moment ago. Some are circular, right? Like the orbicularis oculi, the muscle around the eye. Some are feather-shaped. So here is a muscle, the rectus femoris. And you can tell from even across the room, it kind of looks like a feather, right? Now, sometimes those feather arrangements are like what we call unipennate, half a feather, or a bipennate, meaning that they're equal on both sides, like a regular quill feather. And then there's even multipennate. This represents your deltoid, right? And these pennate muscles are the strongest muscles in your body. because that angle that the fibers come into the tendon creates more power. So your deltoid, right, it's very strong muscle. Your rectus femoris, very strong muscle. Here's the pectoralis major, that convergent, that triangular idea. The rectus abdominis, parallel, right? You see that the fibers are running parallel one to another. And then here's a word I didn't use yet, fusiform. Where else have you seen that term fusiform? That's kind of a fusiform shape like this, right? And fusiform is also the shape to describe smooth muscle cells. So that parasite-looking look, that little wormy look, that's called fusiform. And if you look, the biceps brachii kind of has that same kind of shape, doesn't it? Okay, so that's also considered a fusiform-shaped muscle. And again, if we know these clues within the names, it'll help us. Now, we learned about the trapezius, right? We know it's a trapezoid-shaped muscle. That's where it gets its name. We learned the gluteus maximus, and we learned the gluteus medius. We did not learn the gluteus minimus, but there is one. It's not on our list. The pectoralis major, and there's also a pectoralis minor. We've seen those. The biceps, the triceps. We didn't have any flexor or extensors this semester or pronators. But you get the idea that these names tell us something about what the muscle does or where it's found in the body. Now, in the body, there are over 700 skeletal muscles that are named. We're learning about 45, right? We're not learning that many, to be honest. Every muscle that we're learning in this course in 105 are superficial. They're on the outside of our models. We're not digging in deep, right? In 106, we'll start taking the models apart. We'll see some of the deeper muscles of the leg and of the shoulder, etc. But for now, we're staying on the outside. These muscles, of course, make up your muscular system, and they're scattered throughout our body. Muscles are unique in that they can change shape. So they can move things, right? They can move our digestive products. They can pump our heart. They can help us to move our skeleton. And if you think of a muscle like an organ, just like skin, right? If I gave you a chunk of skin, you could find all 4 tissue types. Same thing in a bone. Same thing in a muscle. A muscle, if I handed you a biceps brachii on a platter, right? It would be largely muscle, but there would also be nerve and epithelial and connective tissues bound up into that thing we call the muscle. We know that skeletal muscle is striated. It has a very strong striping pattern, and by the time we leave here today, you'll understand what's causing that striping pattern. Another characteristic of skeletal muscle cells is that the cells are very, very long. Okay, so if I look at my biceps brachii, this muscle, it is composed of thousands and thousands and thousands of muscle cells. Each of those muscle cells goes the entire length of the muscle. So your muscles are the same length as the muscle cells are the same length as the muscle itself. If you think about the sartorius, right, that muscle that comes around and straps around your thigh, that's a very long muscle. It can be a good 18 inches long or longer in an individual. So those muscle cells are also 18 inches long that make up those muscles. The muscle cell is also called a muscle fiber. So a muscle is made of cells, but a muscle is also composed of fibers. Same term, you'll see it used interchangeably. Finally, as these long muscle cells were formed in your body, what happened is that many little cells connected end to end. And the little nuclei in those cells are maintained. So your skeletal muscle cells are characterized as being striated, yes, but also multinucleated. So they're very long, multinucleated, hundreds of nuclei within one cell. Kind of blows our mind. You know, who's in charge? They work it out. But there's multinucleated cells. Here in lab, you saw or looked at briefly skeletal, smooth, and cardiac muscle. And in the lab review, right, in the mock practical, you should have seen all 3 of those muscle types represented as you walked around the room. In this cartoon, this is a quick way of looking at this. In this cartoon, the top image is skeletal muscle, right? I see the strong striping pattern, strong striations. I want you to appreciate that what you're really seeing here are 3 muscle cells. So, there we go. So from here all the way across. That's one cell that you're seeing a portion of across the entire image. So you're seeing 1, 2 portions of 3 different cells. You can also see that each cell has multiple nuclei, right? So long, multinucleated cells. In the cardiac muscle, I still see striations. There absolutely are striations in cardiac muscle. But what's going to jump out at you, I hope, is or are these intercalated discs. Here it looks like the author used an Etch-a-Sketch, right? Little Etch-a-Sketch lines. But those are the intercalated discs. And we'll talk more about those when we study the heart and the significance of those intercalated discs. But that's really what's going to grab your attention when you look at cardiac muscle. Thirdly, we've got smooth muscle, and each of these cells has that fusiform shape, doesn't it? So each cell has that fusiform shape, and we see that each cell has only one nucleus, centrally located. Other characteristics I could use, do you agree that your skeletal muscle, like your biceps brachii, that it's strong? It's a strong muscle. It can work very, very quick. But it's also discontinuous, meaning I can let my biceps brachii go on break, right? It doesn't have to be working all the time. So it's a discontinuous muscle, and we know that it's voluntary. It works through direct connection with our brain. Whereas cardiac muscle also is quick and strong, but thankfully it's continuous, right? We don't have to wake up and tell it to go. It's going to keep on going through our entire lifespan. But this muscle does not require us to think about it, so it's an involuntary muscle. Finally, the smooth muscle, if you're talking about the tortoise and the hare kind of story, the smooth muscle is definitely the tortoise. I mean, it just kind of moves along, it's kind of weak, kind of slow, but it gets the job done and it keeps on going. So we've got a weak, slow, almost continuous. It kind of just keeps on going, and certainly, like cardiac muscle, is also involuntary. Most of what I'm going to discuss today will be skeletal muscle, but I want to take about 2 slides to give you a review or a mentioning of cardiac muscle. Then I'll give you a couple of slides on smooth muscle. Other than that, everything else I'm discussing today will be skeletal muscle. And every muscle we're naming in the lab is skeletal muscle. So cardiac muscle. I told you that the thing you're looking for will be the intercalated discs, and that is absolutely true. There's the word right there, right? Intercalated discs. But it's also true when you're reading about them or looking at them that you'll notice that they do have a Y branch shape to them, and they are still striated, and they're shorter. They typically have 1, but sometimes they'll have 2 nuclei. Those are all true statements. But again, the key word that you're looking for when describing cardiac muscle will be intercalated discs. Muscle of the heart is also autorhythmic, which means you can take the heart and disconnect it from the brain altogether, and the heart will keep on beating. The heart has within it what's called the pacemaker. We'll see the pacemaker when we get to the heart. We've heard about pacemakers, but the heart has its own autorhythmic pacemaker that tells the heart to beat. And you can take the brain away, and the heart will keep on beating. Right? In a heart transplant, if you watch any of those things on TV, you'll see the heart is still beating. And then they can shock it and start it again without a brain there. It has its own pacemaker. It's autorhythmic. So here's a cartoon of cardiac muscle. Some of this, I mean, again, when you look from across the room, you see striations. Fair enough. Here the artist did not use an Etch-a-Sketch. They kind of used a squiggle machine. So these intercalated discs now show up as these little squiggles. But again, you see them. They pop out at you. I still kind of see that bamboo kind of look, even in a cartoon like this. It does mention Z and A and I bands. For now, don't worry about that. I'll come and mention that toward the end of the talk today, and that'll make more sense to you, but you're not going to have to label anything like that related to the cardiac muscle. Then just a couple of slides about smooth muscle. Again, your major job here is to compare and contrast these. Again, the cells are fusiform in shape, that long, spindly, kind of parasite-looking shape. There'll be a single centrally located nucleus. The next 3 or 4 bullets don't make sense to you yet. They will make more sense to you as you go through this. But smooth muscle, there are no visible striations. So you're looking for the absence, no striations. There are no Z discs, very little sarcoplasmic reticulum, and the T tubules are absent. Those three facts mean nothing to you yet, but they will as we finish up this conversation. When you're going back and reading about this from start again, you will have heard about the Z discs and the sarcoplasmic reticulum in a few minutes. Like I mentioned, smooth muscle is the tortoise, right? It just kind of keeps on going, kind of slow. It just keeps on going. It's resistant to fatigue, right? Your biceps brachii, it wears down pretty quickly. It can't go forever. Your smooth muscle just kind of keep on going. And because they're very slow, they actually don't require as much ATP as well. So they don't require as much energy. So which organelle makes energy? Mitochondria, right? So you would expect to see more mitochondria in skeletal and cardiac muscle than you would in smooth muscle, because smooth muscle just does not require as much energy to do its job. So the smooth muscle is slower to work, slower to relax, and as we know, is involuntary. In this cartoon, you're seeing two cells on the left in a relaxed state, two smooth muscle cells. And we'll deal with more of this in 106. But again, you see this long fusiform-shaped structure. Then, on the right-hand side, the muscle has contracted. It has gotten shorter. It's still kind of fusiform, right? And what you don't see are the same kind of striping patterns that you see in cardiac or skeletal muscle. Okay, everything else I'm going to talk about is going to be skeletal muscle. So we know what this does, right? We know that skeletal muscle is responsible for connecting to our bones and moves our skeleton. We know that it helps to maintain our posture, right? You've got those erector spinae muscles along your spine that are voluntary, that keep you upright. If you lose consciousness or fall asleep on me during lecture, what happens to your posture? You slump over, you start drooling, right? I know when you fall asleep. I can see it, right? The rest of us can see it too. Right? So when you lose consciousness, when you fall asleep, you lose control of your voluntary muscles. This is also the muscle that's going to regulate your temperature. So if you're cold, you're going to get up and move. And as you move, your muscles are using up energy and creating heat, and that heat will be distributed throughout your body through your bloodstream. It's also storing stuff. Here, I want you thinking about your digestive system. So you just had a meal, and that food is going to go down to your stomach and then down through your intestines. And each way along the digestive system, there are sphincter muscles, circular muscle groups, that are going to allow the passage of molecules from one area to the next. And so we've got a lot of sphincters. Now, I just told you this was going to be skeletal muscle. There are two sphincters involved in your digestive system that are, in fact, voluntary. Right? We open our mouth. This is a sphincter. This orbicularis oris, right? We open it up as first part where food goes into our digestive system. And then the anus is also a sphincter muscle over which we have, hopefully, control. The ones in between, though, the sphincters are smooth muscle. Right? We don't have control over those, but they nonetheless are important for moving materials through our body. Finally, not everywhere do we have bones. So your gut muscles create sheets of protection for your abdominal pelvic organs, and there's no bones attached, but the muscles do attach one to another. Speaking of that, what attaches a muscle to a bone? A tendon, right? What attaches a bone to a bone? A ligament, right? Now, what attaches a muscle to a muscle? These are flat sheets of muscle like you see across the six-pack, the rectus abdominis muscles, or that sheet of tendon across the top of the skull. We call that instead a aponeurosis, right? Aponeurosis. And that's a flat sheet of tendon. Connects muscles to muscles. That word was on the last presentation too, wasn't it? Remember there was one slide, tendons, ligaments, aponeuroses. Some of you are looking at me like, that's a new word. I better write that down. Nope, you've seen it. All right? So now you get it written down twice. Now, if I were to ask you, what are the characteristics of epithelial tissues? What would you tell me? Epithelial tissues, Dr. Macauley, are described as having tightly packed cells, right? Forming sheets, layers that sit on the basement membrane. Usually don't have any blood flow going to them. Right? We got that list in our head. If I asked you what are the characteristics of connective tissues, you would instead tell me loosely packed cells surrounded by ground substance and protein fibers. Okay? If I asked you what are the characteristics of muscle tissue, here are the 4 words you're going to tell me. Muscle tissue is excitable. That is, it responds to the nervous system. When the nervous system sends an impulse, it gets excited. We'll talk about that excitability in great detail next semester. When the muscle gets excited, it contracts, right? So it's contractile, or it has contractility. It shortens, it pulls. It can pull bones and change the movement of your skeleton. But also, when a muscle does its job, right? When you think about your biceps brachii, and your biceps brachii is flexing your arm, all the strength of your biceps comes only as the muscle is shortening, as it's contracting. And once the muscle has done its maximum contraction, that's it, right? Now the biceps brachii has to be returned back to its original place, doesn't it, to do the work again. That return to original place is referred to as elasticity. So the muscle also has to be elastic. It has to return back. Just like a rubber band returns back, it's elastic. It goes back to its starting point. The 4th characteristic of skeletal muscles, though, and this one gets a little bit confusing, is that they are also extensible. So as I am squeezing, contracting my biceps brachii, what other muscle is kind of going along for the ride and is getting extended? The triceps, right? The triceps brachii is actually being extended. So we say that skeletal muscles are also extensible because most of our muscles work in opposite pairs, don't they? The quadriceps femoris muscles work opposing or antagonistically to the hamstrings. So when one is contracting, the other one is being extended, isn't it? Right? And then we talk about returning back. That's the elasticity portion. So the 4 characteristics of muscle: excitability, contractility, elasticity, and extensibility. You know from lab that muscles usually don't work in isolation. They usually work in groups. There are really only 2 groups that we've discussed: the quadriceps femoris group and the hamstring group. Let me just clarify again. There is, as we go through this muscle chapter, as we went through the bone chapter, you need to know the bones, the bone markings, the muscles that were presented to you in lab, and that's it, right? I've got too many students who are still like, I'm not sure which muscles to know. Really? It's the muscles on that SOMSO list, right? The muscles of the upper limb, the muscles of the lower limb, the muscles on the little man, the muscles on the big man. What bone markings do you need to know? That 2-sided list that went from the foramen magnum down to the radial tuberosity on the backside. Right? Those are the only bone markings you need to know. Those are the only muscles that you need to know. You need to know all the bones except the carpals and the tarsals, but you do need to know the talus and the calcaneus. Right? So that's the list, right? And then there are 15 tissues you need to know. 6 epithelials, 6 connective, and 3 muscle when it comes to the lab exam. So I'm not asking you to know anything extra or beyond that list for this lecture exam either, except I did add 2 structures in the knee last time? ACL and the PCL. Other than that, right? I got an email the other day. Do I need to know all the structures on the ligament pictures you showed me? No, right? All I did was go through those joints and tell you the bone markings that we already knew. But all the other stuff is extra that you're not at all responsible for. So we know that muscles work in groups. The muscle that does most of the work is called the agonist, also called the prime mover. Now, in the quadriceps femoris group, which muscle is the agonist? Think about it. Which is the most central and largest muscle of the quadriceps femoris group? The rectus femoris, the one big in the center, right? Rectus femoris. So we got the rectus femoris. That means that the 3 vastuses would be considered what? Next word down, they are synergists. They're working with the prime mover. They're working with the agonist, right, to do the work of that muscle group. Quadriceps femoris, right, that's one group. What's antagonistic to the quadriceps femoris group? Hamstring group, right? So the hamstring group is antagonistic, doing the opposite work. What does the hamstring group do for your leg? Overall, those three hamstring muscles do what? Flex, right? They're flexing the leg. What do the four quadriceps femoris muscles do? Extend, right? So make sure you've got that down because those are part of that list of 6 starred muscles that you need to know origin and insertion and action for, right? So the rectus femoris was on that list, wasn't it? And the biceps femoris was on that list. The biceps brachii, the triceps brachii, the sternocleidomastoid, and the gastrocnemius. Those were the 6 muscles that were in that supplement pages that had little stars next to them. And make sure for the lab exam you've looked at their origins, their insertion, and their actions. It's getting really quiet. Hopefully that makes sense to you. Now, that's all largely review, right? Maybe a review of some things you've heard about in lab, things that kind of make sense to us. Nothing horribly brand new there. What I'm about to go into, though, is new when it comes to muscle. And this conversation did not get taken care of in lab, although you did label some of these words and structures in your pre-lab, but we haven't talked about them since. So again, if I handed you a muscle, then you would find within it, right, nerves and blood vessels and connective tissue and everything else. And you would note that the muscle had fibers running in a particular way. Those groups of fibers are said to be in fascicles. We know that term fasci, that prefix, F-A-S-C-I, right? Fasci, fascicle, means a band or a bundle from our vocabulary list. So what you have is within a muscle, you've got these bands or bundles of fibers traveling together. And each of those groups is a fascicle. If you go back to that slide where I said muscles come in different shapes, it says on there the fascicles are in different arrangements. The fibers are running in parallel or pennate or convergent or circular arrangements. When you look inside a muscle cell, it's not going to look like the cell you labeled back in chapters 2 or 3. This cell is going to be chock full of a different organelle called myofibrils. Myofibrils. That's a word you're going to hear me say a lot. Okay, myofibrils. So when you look inside, you're going to see myofibrils, and those myofibrils are themselves made up of myofilaments. Now those two words, I'll say them over and over. They're going to be confusing for a moment, but we'll get them figured out. If I gave you a chunk of muscle, you would also find 3 distinct layers of connective tissue woven within the muscle. These 3 words are not difficult. There is the endo, the peri, and the epimysium. You'll hear the mycelium as well. Mysium or mycelium, same thing. So let's start at the very bottom. Epimysium. Epi, what does epi mean? On, right? On or upon. So the epimysium is on the entire muscle. It's a layer of connective tissue that surrounds the entire muscle. Then there's the perimysium. We haven't gotten to P's yet, but we know perimeter means everything around, right? So the perimysium is a layer of connective tissue around, not the entire muscle, but these fascicles. And I'll show you a picture in a moment. So peri, around what? Around the fascicles. And then endomysium, we know endo means within, right? So this is a layer of connective tissue deep within the muscle, and it's surrounding each and every individual muscle fiber. Okay, so the endomysium is going around every single one of the muscle fibers. Now, in between, right, also traveling through the muscle, in these layers of connective tissue, you would find blood vessels and nerves. Right? Blood vessels and nerves are also traveling down into the muscle tissue through these layers of connective tissue. So here's a picture that I want you to know well. This is a picture you will very likely see on Thursday as one of the images I would want you to be able to label, because it's very straightforward and it's very clear even from the back of the room to figure out what's going on here. So let's take a look. You saw this picture, one like it in lab. So what bone is this? Let's start with that. Do you see the femur? Right? And let's just talk about why we know it's the femur. I see a big rounded head of the femur, and then the narrow region is the neck. This big bump here is the greater trochanter, and this little bump right here, button, is the lesser trochanter. Right? So those 4 of your bone markings on the femur. Let's also talk about what we're seeing on this image for tissues. So what am I seeing at the end? This white stuff is what? What's that white stuff around the head of the femur? Hyaline cartilage. Good. Or you could say, you could be labeled as articulating cartilage, with the cartilage found at the joint space, in the articulation space. And then, of course, this is bone, right? Got that. The tendon is made up of what? Same old thing it was 5 minutes ago, right? Dense, regular connective tissue. And what I want you to see is that the tendon is connecting muscle to bone. And as the tendon comes out, there's a layer that's called the deep fascia. Okay, again, there's that fascie, a bundle, a band, something that goes around the entire muscle. Now, the deep fascia is in addition to the other 3 layers that I've already talked about. So the deep fascia is around the entire muscle. Now, let's take a look at these other 3 layers. We're going to take a look in this box. So we've got this box, and we're now going to blow it up into this bigger area. On the outside, right, where my pointer is, where I'm coloring right now, that outer layer would be called the epimysium. Right? It's the layer surrounding the entire muscle. Again, it's in addition to the deep fascia. Then, if we look inside muscle, we will be struck that the muscle is made up of bundles. Each of these bundles, right, there's a fascicle, there's a fascicle, there's a fascicle. That's a bundle, a fascicle. And each fascicle is surrounded by a layer of connective tissue called the perimysium. So that means in this picture that this thing that's sticking out right there is a what? That's a fascicle, exactly. And that fascicle would be surrounded by perimysium. Now, if that fascicle is a bundle of muscle fibers, then what is sticking out right here? That's a muscle fiber, aka muscle cell. So we see one single muscle cell sticking out here. Remember I told you that muscle cells are as long as the muscle is. That's even proper English, right? But they're very long. And even in this picture, I can appreciate they've even drawn 2 little nuclei in here, 2 little purple nuclei. That muscle cells, skeletal muscle cells, are multinucleated. And I can even appreciate on this slide that there's some striping patterns going on, right? I see some sort of striping patterns going on. So when we think about skeletal muscle, we're thinking about striations. So we even see that in this drawing. So make sure you can appreciate this. So that means that each of these little individual muscle cells is surrounded by a layer of endomysium, right? Endomysium. I'll go through this with you about 3 more times. It'll keep making sense. So here's what I said in words. If you didn't catch it all, it's all here for you. So the epimysium, where is it again? The epimysium is covering the entire muscle. It's going over the entire muscle. Then there's the perimysium. Peri meaning around. Around what? Going around the fascicles. And then finally there's the endomysium within the muscle going around each and every individual cell. So those are 3 layers of connective tissue found within a muscle. So again, I need you to think about this in a more holistic way, that if I just hand you a muscle, it's more than just muscle, right? There's connective tissue and nerve and epithelial. If I hand you a bone, it's more than just bone. There's also in that bone some connective and muscle and epithelial tissue. If I hand you a chunk of skin, right, it's not just epithelium. It's also connective tissue, muscle, and nerve. So keep that in mind. So I'm going to show you the same thing again. We'll believe it if I tell you enough times. So what muscle is this? Sorry, not muscle. What bone is this? Take it back. What bone is this? Yeah, the humerus. Now, why did you not say the femur? Yeah, it doesn't have that big rounded head, right? I mean, it's a much more, it's not quite as crazy of a big head. Now, the humerus has a bump on it up here called the what? The greater tubercle, right? The greater tubercle. So that's on your bone marking list. And once again, I see the tendon, right? That cord-like structure that's connecting the muscle to the bone. And I also see that the tendon is continuous with a layer called the deep fascia. In addition to the deep fascia, I'm going to have sitting on top of the muscle this layer called the epimysium. You can see how they've actually kind of pulled this back. So that's the epimysium, the outer layer on the entire muscle. If I look inside the muscle, I see that the muscle is composed of fascicles. And each fascicle is surrounded by a layer called the perimysium. Okay, oops. And then, if I'm looking, right? So there's a fascicle, and that this would be a fascicle. So that means that the thing that's sticking out is a what? Yeah, that's the muscle fiber. And that muscle fiber would be surrounded by a layer called the endomysium. Okay. Now, there's something extra on this slide that I've said the word, but I haven't had you label it, and I want you to add it for now. It'll make more sense in a moment. These structures that are sticking out down here, at the very bottom, they are myofibrils. Okay, myofibrils. We'll talk about them in a moment. I told you that muscle cells are filled with these things called myofibrils. That's the first picture we've seen of them. Now, the other take-home lesson from this slide, and you know this, you've been around anatomy long enough to know that wherever there is a deep fascia, there must also be a superficial fascia. Right? We don't name major without a minor. We don't name maximus without a medius and a minimus. So if there's a deep fascia, there must also be a superficial fascia. The good news is you already know this, but by a different name. The superficial fascia is the same as your hypodermis. It's that fat layer underneath your skin. So your hypodermis, a.k.a. subcutaneous layer, a.k.a. superficial fascia. Same exact thing. And let me help you make more sense of this. So we're going to slice the thigh. And how many muscles do you need to know in the leg? Quite a few, aren't there? Right? Let's think about it. There were the 3 hamstrings. There's the 4 quadriceps femoris muscles, and my fingers don't count up, right? So 3 hamstrings, 4 quadriceps femoris muscles. There were the 5 muscles of Gapus, right? Gracilis, adductor longus, right? With that one, pectineus, iliopsoas, and sartorius. And then there was also the iliacus and the psoas major coming down from the leg. And then lower, there was the tibialis anterior and the gastrocnemius. That's about 14 or 15 muscles that you need to know on the leg, right? You need to be able to know those on the leg or on the little man or the big man, whatever muscle you're looking at, whatever model you're looking at. So as you cut through the thigh, right, there's a lot of muscles. And we're seeing the femur right in the middle. It looks like a nice steak, right? Nice big old ham steak or something, right? So big old bone in the middle and all these different muscles. You don't have to label those muscles, but you appreciate there's a lot of muscles going through your thigh. And each of those muscles is separated by a layer called the deep fascia. So these muscles can slide over each other, right? So within your thigh, as your hamstrings are doing their job, the quadriceps femoris muscles are kind of sliding in the opposite direction, right? So they're sliding over each other because of that deep fascia. Then in this picture, the superficial fascia is just this layer of fat that would go around the thigh, right? My picture, a little bit bigger layer of superficial fascia, right? But we get it. It's just that layer of fat that's going to go around your skin. So if I had a hypodermal needle right now, and we're going to go from the skin into your muscles, talk to me about the layers. Outside, the needle would first have to penetrate through the epidermis, right? And you know there are 4 layers there, but we won't go there right now. The epidermis. And then deep to the epidermis, there would be the dermis. And then the hypodermis, aka superficial fascia. And then I would hit the deep fascia of a muscle, wouldn't I? And then I would go deeper into the muscle tissue itself. So be thinking about those layers from outside or inside out that you would have to penetrate through with a needle. So I've got about 4 pictures here, and each of these pictures is fully labeled. On the exam, you may see a picture like this. There will always be 1 or 2 clues in the picture telling you where you are. Because after a while, this looks like a bunch of straws stuffed inside of straws, stuffed inside of straws. So this picture tells us that it's a muscle, it says that it's a muscle fiber, right? So we're looking at a muscle cell here. If this was not here, I also know it's a muscle fiber because it's showing me this outer layer peeled back as the endomysium. So either one of those tells me that it's a muscle fiber, right? This is the cell itself. And for the first time, if I look inside the muscle cell, I'm going to see all of these long straw-like structures called myofibrils. So here's the first time it's actually labeled for you on a picture. I can see that within this muscle, there are many nuclei. See all the purple nuclei, right? Multinucleated cell. And there's another cell here, and it's labeled later on, but this yellow guy is a satellite cell. It's coming up labeled, but a satellite cell, right? It's outside. It's in the perimeter of the muscle. If you damage your skeletal muscle, it has the ability of growing back. It has the ability of repairing itself. These satellite cells are stem cells for muscle. So they're there to regenerate if needed through damage. However, your cardiac muscle doesn't have satellite cells. So if you damage your heart, it doesn't respond. It doesn't come back. It doesn't grow back, right? So that's why heart attacks are so devastating, because that muscle doesn't regenerate. But skeletal muscle will. You damage your skeletal muscles, and they have a really good chance of regenerating because of those satellite cells. Let's take a look at this picture and get our orientation here. Take a moment, and there's a couple of key things that are labeled here for us. It tells us that this is the muscle cell. Right? So that means that each of those little guys inside is a what? Myofibril. Then a whole bunch of these muscle cells collectively create a fascicle. And that fascicle would be surrounded by perimysium. Same thing over and over, just different views of this. Let's zoom out 1 layer further. So again, it tells me that this is the muscle fiber. Sorry. So we've got the muscle fiber. That's what's sticking out. And many muscle fibers make up a fascicle. And all the fascicles together make up the entire muscle. And that muscle would have on its outside the epimysium. Now, in addition to the epimysium, there would also be this layer. What's this layer? The deep fascia. Good. And the deep fascia would then be continuous with this tendon, this cord-like tendon, right, that's going toward the bone. So we'd have tendon way back here, and we would have deep fascia here. Does everyone have their orientation okay with these images? If this were middle school or high school class, I'd probably send you to McDonald's and get some straws. Those big old straws McDonald's has, right? And then go over across the street to Burger King and get some smaller straws and stuff some of those straws inside the big McDonald's straw. And then maybe go get some coffee stirrers and stick those into the little Burger King straws inside the McDonald's straws. And then maybe take some rubber bands and put them all around the whole thing. And you'd label it, right? So you'd have this straws within straws within straws within straws. You can kind of think of it that way. And you just have to keep your orientation down, right? What am I looking at in each of these layers? So we know that tendons connect muscle to bone. They're usually cord-like structures, and there's that word again. But sometimes tendons instead are more of a flattened sheet referred to as an aponeurosis. And you see an aponeurosis, none of them you had to label in the lab, but you did see the aponeurosis across the rectus abdominis muscles, that white layer, and you also see a white layer across the top of the skull. That's a muscle-to-muscle connector. Okay, let's practice this. So on the top picture, I know what it is because it tells me, right? But if the word fascicle wasn't there, and if it didn't tell you that this bundle of straws was a fascicle, then what else tells you that it is a fascicle? Yeah, so the layer on the outside is the perimysium. So if you see the perimysium, you know you're looking at a fascicle. That means that each of the things in here is a muscle fiber. Notice that the nerves and arteries and vessels and blood vessels are traveling through those connective tissue layers. On the bottom, if it didn't say muscle fiber, you would know that it is the cell because you see the endomysium being pulled off. Again, these straws coming out of the cell are the myofibrils. These other terms on here, I know they're small. They're going to come up on a slide in a moment, but I want to at least introduce them while we're looking at this image. Muscle cells are just very unique cells. There are no other cells quite like muscle cells. And so individuals have gotten together and said, you know what, they're so different, we have to have a different name for them. So on this slide, you see the term sarcoplasm. Sarcoplasm sounds a lot like cytoplasm. And we're only going to go about 5 slides over, and you're going to see this. So yeah, go to the next page, on the bottom of page 155, and you're going to see this term written out, right? It says sarcoplasm. You see it?
165.
165? Okay, I'm old, can't see it. Right, one page over, on the bottom, you're going to see a slide that says sarcolemma and sarcoplasm, right? So I'm just kind of showing you a picture of those. The words are over there for you on the next page. So the sarcoplasm sounds like cytoplasm. That's exactly what it is. It's a special name for the cytoplasm of a muscle cell. You're going to see 2 prefixes throughout this unit. Myo means muscle, and sarco means muscle. They both are scattered throughout this vocabulary. So myo this and myo that, sarco this and sarco that. Muscle, muscle, muscle. The other term here is sarcolemma. In our vocabulary, we just saw lemma means a rind or a peel-like structure. This is the fancy name for the cytoplasm. Sorry. Plasma membrane. Take it back. This is the fancy name for the plasma membrane around the muscle cell. All right, special name for that rind or peel that goes around it. And then there is that satellite cell that's labeled for us for the first time. In lab, we also spent some time talking about origins and insertions, right? You're responsible for knowing 6 of these specifically. And as you went through that list of, you did 10 of them in your pre-labs. And then in lab, we told you there were 6 to know, right? The 6 starred ones. And what was the overall idea here? Somebody tell me, why do we care about a muscle's origin and insertion? Any takers? Yeah, I mean, every muscle has a unique job to do, right? And the job that a muscle does is related largely to the bones to which it's connected. So every muscle has a job. As that muscle shortens and contracts, it's going to pull part of the skeleton. The origin is defined as the less movable place. If you look at that list, those 6 muscles, if you look at, for example, the biceps brachii, it's going to tell you that there are 2 tendons up into the shoulder, and one is going to connect to the coracoid process, and one to a portion of the glenoid cavity. Right, so we've got 2 tendons originating up in the shoulder for the biceps brachii, and then the biceps brachii comes down and inserts into what? What bone marking on your radius? The radial tuberosity, right? So that becomes the insertion place for the biceps brachii. And so it makes sense, right? When the biceps does its work, it's pulling not only the radius, but also the ulna, right, up toward the shoulder. By definition, the less movable place is the origin, and the more movable place is the insertion. Okay. So we saw those ideas, and in this image then, Even the drawing shows me that the biceps brachii is a two-headed muscle. I see one tendon going up to the coracoid process. I see the other going around to the glenoid cavity. And it goes down and inserts onto the radial tuberosity. Right? So up here is the origin. Down here is the insertion. And every muscle has such places where it attaches, and we want you to know the specifics for 6. Right? Just for 6 of them for the lab exam. If you're going to physical therapy or med school, you would learn the origins and insertions for probably about 250, maybe as many as 350 of the muscles. So you would know exactly where they attach, the origin, the insertion, what the muscle does. You would know which nerves bring information to that muscle. It is a very large, what I would call hideous body of knowledge. It's just huge, right? Page after page after page of origins and insertions and actions. We want you to know 6. We think you can handle this. Now, as you look at all the drawings, and you're looking at these layers of the endo and peri and epimysium, you'll also see scattered within those images red and blue vessels, blood vessels, and yellow nerves. A reminder that every muscle is made up of hundreds or thousands of cells. And every one of those cells needs oxygen, right, and nutrients. So we have to have oxygen and blood vessels going to it. We need to get rid of waste products. So we have veins carrying the waste products away. And there's also going to be nerves going down and directly connecting to every single muscle cell in the muscle. So we have to have a way, right, a pathway for arteries and veins and nerves to reach the cells. And so as you see the drawings, you'll see those vessels and nerves drawn throughout those connective tissue layers. Like I mentioned, muscle is very, very energy needy. Got to have a lot of oxygen to make a lot of ATP. We also are building up waste products. If you're really working your muscle, we know what happens, right? Your muscle fatigues, but also what builds up in your muscle? Lactic acid, right? We'll talk about that story in another class, but that's a waste product that must be dealt with, and that waste product has to be carried away by vessels. And then finally, you've got to get oxygen to your tissues as well. So skeletal muscle is voluntary. It only contracts when you tell it to. You have voluntary control over it. Now, the nerves that connect to your muscles, right, that go from your brain down your spinal cord and out to your muscles, those particular cells that tell the muscle to contract are called motor neurons. I'm not going to talk much about neurons at all this time, but these are the cells that literally go from your spinal cord down to each and every one of your muscles, and they're referred to as your motor neurons. When you hear the word motor, just think movement, right? And the motor neurons are carrying signals to your muscles telling you to move. We will talk about the nervous system more than any other system this semester. We're going to have 5 chapters on the nervous system. When we get back from the test next week, we'll start talking about neurons and how the nervous system works, and then we'll have a whole chapter on the spinal cord, a whole chapter on the brain, and a whole chapter on the autonomic nervous system. When we're all done with that, we'll have a whole chapter on the special senses like sight and hearing and vision. So we've got a lot coming up related to the nervous system. So I just want you to appreciate that every muscle cell is directly connected to a nerve through what's called a motor neuron. And that's it for now. And when a muscle receives that electrical signal from the nerve, it contracts, right? That's what it does. It's excitable. It receives that signal, and as it becomes excited, the muscle contracts. That's what's going on between the brain and the muscles. Here are those terms, right? I pointed them to you already, so let's just make sure we see them, and actually I'm going to cross off a couple of these right now. So the sarcolemma, again, special name for the rind, the peel, the outer layer of a muscle cell. You would call it the plasma membrane of a muscle cell, sarcolemma. Sarcoplasm, just a specialized name for the cytoplasm of a muscle cell. T-tubules, we're going to cross it off. I'm not going to even mention T-tubules. Again, this semester, we'll deal with those much greater in Biology 106 when we take a look at at the molecular level what is going on as muscle contracts. But I'm going to skip over that right now. And then I'm going to mention this word sarcoplasmic reticulum. Sounds a lot like what? Endoplasmic reticulum, and even more like smooth endoplasmic reticulum. And that's really kind of what it is. What does smooth endoplasmic reticulum do in most cells? The smooth endoplasmic reticulum, recall, was making lipids and steroids. Well, muscle cells don't need much of that. What muscle cells need is a lot of calcium. So the sarcoplasmic reticulum is a specialized organelle found only in muscle that stores calcium. And I just want you to remember that calcium is hugely important for muscles to work. I mentioned that back in skeletal land, right? I said, you know what? Bones store calcium, and one of the things that our body needs calcium for at high levels is muscle contraction. I mentioned the nerves to work properly, and I mentioned blood clotting, right? So I want you to see that calcium is a critical player within the contraction of muscle. It is stored in the sarcoplasmic reticulum. Kind of just keep that, it's like a little silo, a little storage place of calcium, and you'll understand that better next semester.
So how does potassium play a role when you have a muscle spasm?
Potassium also plays a role. Potassium and calcium tend to kind of work in yin-yang a little bit, and potassium definitely can cause muscles to, or at least keep them from contracting as much, right? Relax a little bit. Yeah, eat a banana, right? Potassium, help out with muscle cramps. Okay, so we've looked at the fascicles and we've looked at muscle cells. Now we're going to dive in just a little bit deeper into the muscle cell. Not a whole lot more, but a little bit more. And within the muscle cell, that is within the sarcoplasm, right? Within the cytoplasm of a muscle cell, I'm not going to see what I usually see in a cell. I instead am going to be struck by hundreds to thousands of these things called myofibrils. Again, it just looks like a bunch of straws stuffed inside of the muscle cell. And each of those myofibrils is actually composed of shorter myofilaments. We're going to get this. Now, myofibrils run the entire length of the muscle cell. So let's get a picture here. I told you that for my biceps brachii, that the cells run the entire length of the muscle. Within the muscle cell, also these myofibrils run the entire length of the muscle cell. So they're very long, these myofibrils. And the myofibrils are made up of shorter units called myofilaments. Now, where have you heard about myofilaments? Maybe not myofilaments. Where have you heard of the word filament? Until a few years ago, where could we find filaments in our home? Light bulbs, right? That little tungsten filament, that little wire, that very, very thin wire, right? In a light bulb. We've all seen it. So when you hear the word filament, think very thin wire. I mean, very, very thin structure. Okay. Now, it turns out that these filaments in your muscles come in two flavors, thick and thin. Okay. So we'll start off with the thin filaments. Now, on the top of this slide, put myofilaments, right? Because this is a myofilament. This is a filament of muscle. There's two types, thin and thick. The thin filament is actually composed of a protein called actin. Okay, and I'll show you a picture of this, and in a moment you're going to see that these actin molecules kind of make like a helix. They make a little double helix. I'm going to have you cross off tropomyosin and troponin. I'm not going to ask about it, don't need to know about it. We'll do that next semester. So just know that actin is a little helix. It kind of looks like a bunch of little strings on a beads on a string. And finally, actin is going to bind this other molecule called myosin. That's all you need to know about actin right now. The other myofilament is the thick filament, and the protein here is myosin. Myo meaning muscle, IN, it's a protein, right? The major muscle of the major protein of muscle. It's bigger, so it's called the thick filament. It binds to the actin. And I'll show you a picture of this in a moment. And what we're going to see is that when you look at skeletal muscle under the microscope and cardiac muscle, you see the striations. And those striations really are an overlapping pattern of these actin and myosin molecules. So what's causing that striping pattern is the overlapping pattern of the thin and thick filaments. Let's take a look at a cartoon of this. On the bottom, that big yellow molecule, that's the thick filament, that's myosin. Okay, it's actually a bunch of myosins together, but we see myosins. Some people describe a myosin as being like a golf club, right? It's a long protein, and it has this globular head that kind of looks like a golf club. Okay, so there's the golf club. So you see all these golf clubs. When I see myosin, if you've ever seen Velcro under a microscope, that's what I think of. Something that grips, right, and kind of holds. All those little fingers grabbing onto the Velcro. That's what I kind of picture when I think about myosin. Then up here in red is the actin. So you see one layer of red and another layer of red. Ignore the green and blue. Right? Those are those other things I told you to ignore. So the red, you just see the red helix of these actin molecules. On the top, this straw, right, for the very first time we're seeing a myofibril in isolation. That myofibril, remember there would be hundreds or thousands of those myofibrils stuffed into the sarcoplasm. And for the very first time, we're seeing that the myofibril is itself made up of little tiny straws called the myofilaments. So now these are those little tungsten filaments, the smallest little wire you could ever imagine. Those myofilaments come in two different types, right? Actin and myosin. So that's what those myofilaments are, the actin and the myosin, the thick and the thin filaments. Now I'm going to skip over this slide. I'm going to tell you what to cross off, then I'm going to skip over it, and then we'll come back to it. In muscle, what you want to be looking for is I do want you to know A, I, and M and Z. You can go ahead and cross off H. I'm not going to ask you about H. So cross off H. And I'm also going to let you cross off connectins, and below it, the word titans. Okay? So you can cross off connectins, titans, and H. I will come back and I will tell you about A, I, M, and Z. So A, I, M, and Z we'll talk about here in a moment. Let's take a look. On the top, we've got a cartoon representation of a very, very small part of muscle. I mean, we're down now way below what you can see on a light microscope. In fact, the bottom image is from an electron microscope. When you look at muscle, you're struck by that tire track pattern, right? That striping, striation pattern. So I see alternating areas of dark and light down here in this electron micrograph. Well, let's figure out what's going on here. So the yellow, same colors, the yellow molecule right here that's a myosin. Okay? That's a thick filament. The red guys over here, there's a thin filament, and here's a thin filament. Wherever I see the yellow myosin, it's a pretty big, chunky protein, right? So wherever I see the yellow myosin, I see a darker area on the micrograph. It's a thicker protein. Wherever I only see my actins over here, right, they're thinner, there's not as much to them, and so I have lighter bands. So what I'm seeing when I look at striations of skeletal muscle under the microscope, I'm seeing an alternating light and dark pattern based upon the overlapping actin and myosin myofilaments. Okay. On this, just to get our bearings, this whole thing, this whole little section is called a sarcomere. Now it says that it goes from here to here, and that's a sarcomere. What does sarco mean again? Muscle. How about, right, muscle. What about mere? Mere or mer? A polymer is a molecule made up of many parts. A monomer, something made up of only one part. A dimer made up of two parts. So sarcomere is what? A part of muscle. It is the part that is actually doing all the work. We're down here at the microscopic level, and what we're seeing is that these overlapping actins and myosins are going to create the pulling force that muscles can exhibit. A sarcomere, this little unit, is defined as going from Z to Z, from Z disc to Z disc. Okay, I can figure that out because Z is the last letter of the alphabet. So the ends, right? The ends, the book ends of the sarcomere are defined by Z. Z to Z. And I'll go back to those letters in a moment. Then we're in the very, very middle. That's easy enough. In the very, very middle is the M line. Okay, middle, medial, easy, in the middle. Wherever I see just the, I'll clean this, wherever I see just the thick filaments, right? The thick filaments are the same as the A band. The A band is going to be this darker area. Right, so from here to here, that's the A band. A is a wide letter, isn't it? It's a broad letter, thick filament, myosin. The I band is only where I see just actins. Okay, so I is a skinny little letter, thin filaments. So the I, thin filaments, actin. A is a broad letter, thick filament. M in the middle, and Z to Z, the ends of each sarcomere. Those are the four I want you to know. So if we go back and take a look, right? Here they are in words. So Z to Z, from Z to Z, that defines the sarcomere. The M line was right down the middle. The I band is where I only have the thin filaments. The A band is only where I have the thick filaments. Don't worry about H. Don't worry about connectins. Don't worry about titans. So let's get a better appreciation of this if we can. I want you to, this picture's good, but it makes it look like muscles flat, that is two-dimensional. And don't forget that muscle cells are three-dimensional cylinders and muscles are long cylindrical structures. So if we look at this picture, I think we get a more complete understanding. This thing that's sticking out, this is the muscle cell. Okay? And that muscle cell has myofibrils sticking out of it, doesn't it? Right? Lots of myofibrils. And the myofibrils are actually made up of overlapping segments of myofilaments. So as you look at this, there's a Z, and here's a Z. It tells me that that's a sarcomere from Z to Z. And within that Z to Z area, I see areas of dark and areas of light. Right? Overlapping light and dark areas caused by overlapping myosins and actins. Along the length of a myofibril, there will be many myofilament units, many, many sarcomeres. So there's one sarcomere, here's another sarcomere, there's another sarcomere, there's another sarcomere. So there's a sarcomere, 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 sarcomere. Right? Many, many sarcomeres down the entire length of the myofibril. Now what happens is that when the muscle contracts, when the nerve sends its signal, and the muscle receives that excitation signal, the sarcomere shortens. And all of the sarcomeres simultaneously along the length of this thing will shorten. And that's where you get your force. That's where the energy of a muscle comes from. So when someone squeezes their biceps brachii, right, the muscle is shortening. Why? Because each of these little individual microscopic units, called a sarcomere, is shortening. It is shortening because the actins and the myosins are sliding over each other. So there's that word sarcomere again. And again, it's defined as being the contractile unit, the functional unit. It's the real action center of what muscles do. And again, a sarcomere is defined as going from Z to Z. And along a myofibril, there would be many sarcomeres, many of these Z discs. As a muscle shortens, the sarcomere also is shortening. So imagine for a second that my fingers are actins and myosins. It's a pretty decent visual. My thumbs would represent the Z discs. So that means that between my thumbs, that's a what? Sarcomere. And when muscle gets the signal to contract, the actins and the myosins are going to slide over each other. Now, as they're sliding over each other, what's happening to my sarcomere? Got shorter, right? Now, don't forget that the muscle, the myofibril, would have thousands of these sarcomeres linked end to end. And as the signal comes in, all of them would shorten, and the whole muscle would contract and get shorter. Okay? So it's like tug of war. These little actins and myosins are grabbing each other and going shorter, 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 shorter. Now, this is a very high energy process. It takes a lot of energy for your muscles to do this work. Yes, as the muscle contracts, the Z discs are getting shorter. But are my fingers getting any shorter? Right? As the actins and myosins are sliding over each other, the actins and myosins are staying the same length. Right? So my proteins are not changing their shape. Instead, they're just simply sliding over each other and thereby causing the muscle to contract. So let's take a look. This whole idea that these filaments are sliding over each other is aptly named the sliding filament theory. And during this sliding, what happens? We saw that the sarcomere gets more narrow, right? It shortens. And the Z discs, therefore the Z lines would get closer and closer, right? My thumbs got closer to each other. The A band, though, stays the same. Because what did the A band equal? The A band was the same as the myosin. But as I just showed you, my fingers aren't getting shorter. My myosins aren't getting shorter. So the A band actually is not going to change its width. The I band will narrow, and I'll show this to you in a moment. So the bigger a muscle is, the more of these myofibrils it has. And the more myofibrils that a muscle has, the more actins and myosins it has to pull. This is where the energy comes from. A bodybuilder has more myosin, more actin, more sliding filaments, more power that can be generated from movement of the muscle. So here's a cartoon of a relaxed muscle. It's the same picture I showed you a moment ago. So we're looking at just a sarcomere from Z to Z, right? From Z to Z. There's my sarcomere. The darker area is where the myosins are, right? And the lighter areas are where just the actins are. Now what happens as this muscle begins to contract? This picture changes. If you look at the cartoon, now you see that the actins and myosins have slid over each other. They're more compacted. As a result, right, my banding pattern would change in the muscle under the microscope. But the A band did not change. The A band is still going from how wide the myosin is. So the A band did not change. The I bands did change, however, because the areas that are only actins do get smaller. So the I band will get smaller, but the A band does not get smaller. And then when you're squeezing your muscle as hard as you possibly can, what's going on is that all of those actins and myosins have overlapped to their maximum ability. So every single actin and myosin has slid over, shortening every sarcomere as much as it possibly can. All of those sarcomeres have simultaneously shortened. There's nothing more you can do, right? I mean, you've contracted your muscle. There's no further that it can do. And in fact, it starts to lose power too, doesn't it? There's only so long we can hold maximum contraction. Now, I've got 5 minutes, and I'm going to skip a lot of slides here. Okay, question on A, M, I, and Z.
You said that A band is made of
Myosin. So the A band is wherever just the yellow myosin is found. The I band is only where the actins are by themselves. Z to Z is the sarcomere, and the M line is down the middle. So as the sarcomere is shortening, my thumbs are getting closer, aren't they? So Z to Z is getting closer, the sarcomere is getting shorter. But my fingers are not getting shorter. So the myosins, the A band is not shortening. That's where this falls apart, right? This doesn't show A and I very well. My fingers can't, right? But if you look at the picture, you see the A band stays the same. It's the I band that gets shorter on each side. Now, you know that nerves come down and touch muscles. That's going to be called the neuromuscular junction. But I'm not going to deal with any of this right now. So you're going to put a big old X through these slides. The only word that I want you to circle anywhere in this slide, I'm going down to, what slide is this one right here? What number is this? 168. The only word I want you to see on this whole slide is, oh look, calcium is important, right? Calcium, calcium, calcium. I just want you to realize that calcium is necessary for muscles to do their work, okay? We're going to go down. We're going to keep on going. The other word I want you to circle on this word is that ATP is necessary, right? ATP is necessary. Energy is necessary. So you need calcium and you need ATP. And don't forget, also to make ATP, you need oxygen, right? So you need to have oxygen, you need to have ATP, and you need to have calcium for your muscles to work properly. And that brings me to this picture. So we kind of skipped over everything to the cat. I want you to look at this slide on the cat. Okay? Now, no animals were harmed in the making of this picture. I do not know if this cat's sleeping, stretching, or dead, but it looks like it's in rigor. Now, rigor mortis is a state that our body will go into after death. About 3 hours after death, up to about 36 hours post-death, the body will go into a rigid state. Okay, so what's going on? The muscles are in some state of contraction, right? All of your muscles at the time of death will be in some state of contraction. Within about 3 hours after you pass, your muscles will no longer have ATP available to them, right? There's a little bit around for a little while, but ATP gets depleted very, very quickly. And the details of this is that it takes ATP for your muscles to relax. So after death, your muscles go into a state of rigid contraction. There's no more ATP around. And it stays there until about 36 hours later, and then the muscles start to relax. Now, they didn't start to relax because suddenly there's ATP available, but by 36 hours, the body actually starts to break down, and the muscles are relaxing at that point. Okay, so that's rigor mortis. Just know that it has to do with energy availability after death. Then we're skipping the next slides. Nothing about motor neurons, motor units, nothing, 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 all or none, nothing. Nothing on tone, nothing on contraction. Nothing on atrophy or hypertrophy. Nothing about energy or fatigue. I only want you to go down to the second to last slide that says what happens to the skeletal system as you get older. So skip everything down until the aging slide. Right? I'll try to finish up every system with what happens as we get older. Okay? All the way to the end, right before the muscle man. Okay? Aging. So what happens as we get older? Slowly we'll lose our skeletal muscle mass. As we lose our muscle mass, our muscles will become smaller and less powerful. The muscle tissue that we lose will be replaced by fat or connective tissue. Not only will we lose skeletal muscle, but we'll lose cardiovascular muscle. Now, can we slow this down? Absolutely, right? It's not like this is inevitable. If we stay active, we can slow this process down. But not only will we decrease our skeletal muscles, but also decrease our cardiac muscle. That's going to make our heart less efficient. Well, not only are your muscles weaker, but now your heart's not able to send as much oxygen to those muscles. And so overall, you start having even more issues with your skeletal muscles. Your ability to tolerate exercise will decrease. You will fatigue more rapidly. It'll take longer for you to recover from a game of soccer, right, when you're 60 than it will when you're 20. That's pretty obvious. And you're more prone to injury as well. Finally, your muscles become less elastic with age. So that's what we all have to look forward to, right, as our bodies age related to our muscles. And then finally, this picture I thought was photoshopped. And about 4 years ago now, one of my students at MSU at the medical school said, my goodness, I know that guy. He was my middle school PE teacher. So he's not a Photoshop, he's real. John something. He has died of pancreatic cancer. He had a blog out, and basically he obviously shot up with enough steroids, right, to keep a business open. And those steroids killed him. He died very, very prematurely from liver damage from the steroids. And the story in his blog was basically it wasn't worth it, right? He was this big bodybuilder guy. He had this body that looks like it's photoshopped. He wore apparently just T-shirts because who can wear? Kohl's doesn't sell that shirt. And anyway, he got that way through a lot of hard work, but also through a lot of medical enhancements, right? So that's the end of the material for exam number 3. Make sure that you have done your homework assignments on mastering, the 3 homework assignments. Make sure you've also done the quiz. That quiz is due by midnight tomorrow night. So make sure you hit that mastering quiz. And then if you have lab exam, you also have your PALS quiz to be looking at. Now, keep in mind, if you don't have lab until next Tuesday, right, and some of you don't have lab exam until next Tuesday, you might still want to look at that PALS quiz before you take the lecture exam, because that PALS quiz is over bone markings and muscles and bones. And so it wouldn't hurt you to do the PALS quiz and the mastering quiz both before I see you for the lecture exam. Okay, just a possibility as another way of practicing and studying. Come up and see me if you have any questions at all.